Hello everyone and welcome back. We're going to continue where we left off. As you recall, we were talking about different types of macromolecules that we find inside the human body, and we had covered a number of them, including carbohydrates and lipids. And now we're going to cover the last two categories, and that's proteins and nucleic acids. So let's quickly write down a little outline of where we're going to go today. So as I said, we're going to start a discussion on proteins. And under this, we're going to talk about the function of proteins. We will also, look at the composition of proteins. And we will look at the structure, how they're put together, of proteins. And we'll talk about a special type of protein, which is an enzyme. And this discussion is going to be important because throughout the remainder of this course, as well as 40B and 40C, from time to time, you will need to understand, and we will be talking about this protein that we call enzymes and what they do and how they function. So we're going to have an understanding of that, hopefully, at the end of this discussion. And then, once that is completed, we're going to look at nucleic acids. And as you may know, nucleic acids include DNA and RNA. And under this category, we're going to look at ATP. Now, we're going to very briefly talk about nucleic acids because they don't play a very important role in our discussions in the human body, at least not in this class. And so we're not going to really spend a lot of time there talking about them. We'll become familiar with them and then let it go. But we do really want to understand this molecule because that particular molecule plays an extremely important role in all of the reactions and physiology that you're going to end up talking about, whether it's this class or the next two classes that are part of this sequence of classes. All right, so let's begin that discussion. And let's go ahead and start with proteins, as we mentioned. So we'll begin that. We'll head this with that word, proteins. Okay. And one of the things that we can say is that this is a very large and diverse set of molecules. There are many, many different kinds of proteins found in the human body and throughout all of life. Because it is a very large and diverse set of molecules, you must know that this means that there are, they also represent a very large and diverse set of functions. This goes back to what we had talked about in the very beginning of this class. Form follows function. So if you have a lot of diversity in form, you're going to expect a lot of diversity in functions. So we're going to go through and just quickly identify some of the major categories in terms of function that proteins provide for us. So let's get that under functions. Okay. So one of the things that proteins do, or one of the functions they provide for us, is a structural components. Of the body. And this takes the form of many, many different types of proteins. So I'm just going to give you one example. Ligaments. And a ligament is connective tissue that connects bone to bone, and it's composed of this protein, collagen. In fact, this is probably the most abundant protein in the human body. is collagen. And it's not just a single protein. There are about at least 5 different varieties of collagen proteins, and each with their own individual function and behavior. So structural components of the body, for sure. A second function is regulatory. Okay. So this would be things like hormones. So these would be signaling molecules that change the physiology and the behavior of cells in our body, and hormones play a part. And an example of that would be insulin. And we have talked about insulin when we were looking at homeostasis and control of the blood glucose levels. Insulin plays a very important role in that. A third function is contractile. And perhaps a better way to talk about this function is movement. So movement is being produced by proteins. So, and obviously what comes to mind is muscles, whether it's skeletal muscle, cardiac muscle, or smooth muscle. So when we look at, as we will, the last chapter we cover in this class is about muscle tissue, and we'll look at how a muscle moves, how it contracts, and what's involved with that. And what we'll discover is that there are a number of proteins that are responsible for producing that movement. And I'm just going to mention 2 right now, actin and myosin. And at this point in this class, we don't really need to know what these proteins are. They're just examples of what we're calling contractile proteins. They produce movement. And we'll become very familiar with them when we cover the last chapter of this course and we look at how muscles work and what they are, how they produce movement in the human body. So actin and myosin. Another important function, and let me very quickly figure out where I am. So this is number 4. Number 4 is immunological. So proteins play an immunological function in our bodies, and a classic example of that would be antibodies. These are proteins that are produced by your immune system to help you fight off disease, pathogens, and so forth. So a very important function, obviously, and it is guided by, or this reaction, this protection is the result of a type of protein that we call an antibody. Number 5 is transport. Okay. So there are a number of proteins that are acting as transports. That is, they transport different substances within the body. Let me just give you one very good example. This would be hemoglobin. So hemoglobin is a protein, and it is transporting oxygen and carbon dioxide. So a lot of people don't realize that carbon dioxide is part of the story. So what we're looking at is the transport of oxygen in the blood from the lungs to the tissues. And then when the blood goes back to the lungs after it passes through our tissues, it's carrying carbon dioxide. So this is a molecule. It's a protein that's transporting oxygen and carbon dioxide. There are many examples of this that we have in our body. Going back to our most recent discussion, we looked at lipids, and I pointed out why lipids are not soluble in water. And yet, lipids need to be transported in the body, and they're being transported in the liquids that make up most of our body. So how is this possible? How can we transport something which is not dissolvable in water? How can we use water to transport that substance? And the answer is we're going to hook a protein to it, or a protein will bind to that, and that will be the mechanism by which we transport these non-soluble or insoluble substances within our body. You'll learn more about that later on in some of the other organ systems that cover particularly digestive tract. All right, so transport, very important function for proteins. A sixth function is catalytic. All right, so what is a catalyst? And the answer to that question is a catalyst is a substance which speeds up a chemical reaction but does not itself get changed with that reaction. Okay? And we'll write this definition down a little bit later because we're going to talk about the proteins that we call that have this catalytic property, and that would be enzymes. So enzymes are catalysts. They speed up chemical reactions, but they're not changed in that chemical reaction. They simply speed it up. And a very good example of this would be pepsin. Pepsin is found being produced in the stomach, and it helps to break down the proteins that you consume. It is an enzyme. It is an enzyme which breaks down the bonds between amino acids so that the very large proteins that you consume are slowly broken down into their smaller component parts. Okay? And pepsin is playing a role in that, and pepsin is a protein. Pepsin is an enzyme. All right. Well, that's a very brief sort of discussion of the roles. It's brief because when we get into more specific kinds of things, as we will, in fact, in the next chapter with cells, we'll look more closely at the functions of proteins and examine that with some more detail. So right now, we're just getting a general idea of what the function of proteins are in the human body. And we'll go into the details when necessary as it comes up. All right. So functions. The next thing on our list that we need to talk about is composition. How are proteins put together? And what are they composed of? So let's start with the basics. If we look at the elements that the proteins are composed of, we're going to see the same players we had talked about earlier. We're going to see carbon. There's no news there. We've seen that before popping up, and because this is an organic molecule, of course, it's going to contain carbon. Hydrogen, oxygen. So all the usual suspects, as we've seen in both lipids and carbohydrates. So where do we see a difference? The next element really is going to be the key. Nitrogen. So nitrogen is going to be a big player here in the formation of proteins. And if you see, or if you remember back to that chart that we looked at the very beginning in the first lecture in this series, and I'll bring it up right now so we can look at it again, where we're looking at the major elements that make up the human body. Let's go ahead and do that. And let's focus here on the top line there. And as you can see, here's our players: oxygen, carbon, and hydrogen. And nitrogen down here at the bottom, about 3.2%. That is, in all, that number there is the result of proteins. That is why we have this percentage and not these very, very small percentages you see down here for these other elements. That's because it's a major player in the way proteins are put together. So that's why that's on the top of the line and not below that line, is because it is being used to put together proteins. So a protein, that's the basic elements that our proteins are composed of. But proteins are composed of a long chain of amino acids. A long chain of amino acids. That's what a protein is composed of. So those amino acids have carbon, hydrogen, oxygen, and nitrogen, as well as probably some other elements depending on the type of amino acid, such as sulfur. But the protein itself is composed of an amino acid. And an amino acid has 3 parts to it. Has 3 parts to it. Those parts are, number 1, a central carbon molecule or atom. Right. Number 2, a carboxyl group. Number 3, an amino group. And right away I see that I made a mistake. This would be my, let's see, what is it? 3rd mistake this year. This number is incorrect. We need to add 2 more parts to this. So it's going to be 5 parts rather than 3 parts. So please excuse my mistake there. So 5 parts, central carbon atom, carboxyl group, an amino group, and a hydrogen. And then the 5th group is going to be our side chain. And this is going to vary depending on what type of amino acid is involved here. So 5 parts. Let's go ahead and draw a quick picture for our notes of the basic structure of an amino acid, and we'll show you some pictures of that. We'll talk about it a little bit more. So let's begin then with the central carbon atom. So we're going to start off with the C. That's our carbon. Okay. And as you know, carbon has the ability to bond to 4 different things. So there's our central carbon. And over here, we're going to attach this, another carbon, oxygen, and that right there. And this, ladies and gentlemen, is going to be our carboxyl group. And then over on this side, we're going to add nitrogen and hydrogens. And that, ladies and gentlemen, is going to be our amino group. And there you see the nitrogen. That's part of the elements that make up a protein. All right. And then we're going to have right here, we're going to have a hydrogen. And that's self-explanatory. It's just a hydrogen. And then right here, this is going to be our side chain. So that's the basic makeup of a amino acids. Let's go ahead, and I'm going to show you a better drawing than I just produced, just to reinforce what we've done. So here is, we just focus in right here. So here's my central carbon. Here's my carboxyl group. Here's my amino group. There's the hydrogen, and there's my side chain. Okay. So notice what they call this, a non-iodized form of an amino acid, a non-iodized form. So also notice that they're applying this term base here and acid over here. So if you think back in terms of what we defined inorganic acids, an inorganic acid releases a hydrogen ion. So indeed, this will, under the right circumstances, release a hydrogen ion. And here you can see in this picture, so this is the ionized form. We've lost the hydrogen right here. So that makes this oxygen a negative. Okay. And then on this side, they're calling it a base, which means that we can attach an extra hydrogen. We can soak up, if you will, a hydrogen from the solution and attach it to this nitrogen. And that's what you see right here in this image right here. So there's my extra nitrogen, which makes this side of the amino acid positive and this side negative. And now I hope you know that if I have this situation, this thing can be dissolved in water because it has a charge associated with it. Okay? So this has a polar bond here, so there is, to some degree, an ability to dissolve this, but this is very easily dissolved in water because of those charges that you see right there. So that's your basic amino acid. But let's turn our attention to this area right here, the so-called side chain, which they're marking in R. What is that, and what happens there? Well, we can create 20 different amino acids, and it turns out that our bodies utilize 20 different amino acids to produce a variety of different proteins in our body. So each amino acid, each different 20 amino acid, or each amino acid that's different within that group of 20, is different because there's something here which is different than all the others. And let's look at some examples. So here, for example, is glycine. And all we did was added another hydrogen here. And that makes this amino acid glycine. If you look over here, we've changed that. It's no longer a hydrogen, as you see here. As you can see over here, it is now this side group here. This is sulfur. And we have cytosine. So by simply changing the side group from one type of molecule or another, I can produce 20 different. Here's lysine. Here is tyrosine. Okay. So there's the variety. There is how we get variety. 20 different amino acids by changing the side chain here. So there are 20 different side chains that produce 20 different amino acids. Okay. All right. So let's get that into our notes. So there are 20 different side groups producing 20 different amino acids. We are composed, or I should say our proteins are composed of 20 different amino acids. So the building blocks that make up our proteins, our entire building set of building blocks consist of 20 different blocks, 20 different amino acids. We put those down in different combinations, and we get different types of proteins. Now, I just want to return, I'm going to pick up on this in just a second, but I just want to return back to that drawing, that image that we saw here, and I want to point out something which I didn't emphasize the first time we were looking at this. So non-ionized and ionized form of an amino acid. I already talked about the significance of that means that this can be easily dissolvable in water, but it can also act as a buffer, and we are not going to define what a buffer is right now, but very quickly, a buffer is a solution that can control, or is a molecule, I should say. It's not a solution. It's a molecule that can control the pH in a solution. So if, for example, the pH goes down in a solution, meaning that it becomes more acidic, what will tend to happen is this hydrogen will tend to attach itself to the nitrogen. And so those hydrogen molecules will be taken out of solution, returning that solution back towards its normal pH. On the other hand, if a pH is becoming more basic, meaning that the pH scale is increasing above 7, then what this molecule can do is release a hydrogen here, as it did here, and thus control the change in pH in any given solution. And you're going to talk about this concept in 40C when you're doing electrolytes, the chapter on electrolytes in this textbook. So this is going to be one of the things that you're going to run into is proteins as buffers in the body. Okay. All right. So as we said, 20 different types of amino acids, and we're hooking them up in chains. So let's talk about these chains. Let's talk about the terms to describe different chains of amino acids. So if I have 2 amino acids, what I'm going to call that is a dipeptide. Dipeptide. If I have 3 amino acids, you can probably guess this, it's going to be a what? A tripeptide. That is correct. If I have 4 to 10 amino acids, then what I'm talking about is a peptide. And to me, this is a very unfortunate term to use. It can be very confusing, a peptide. I wish they had changed it to something else, but there you have it. We have to go with it. So 4 to 10 amino acids, a chain, a protein chain of 4 to 10 amino acids is referred to as a peptide. And more than 10 amino acids is a polypeptide. So more than 10 amino acids and we have a polypeptide. So why all this peptide talk? The answer to that is that amino acids are held together by a very special type of bond called a peptide bond. Let's take a look at an example of what I'm talking about here. So in this drawing, I have glycine and alanine, and I want to hook them together. And as you can see, I'm going to remove water. So there is that dehydration synthesis reaction again. As I said before, this is going to pop up over and over and over again, dehydration synthesis. So I'm going to remove this hydroxyl group and that hydrogen. I'm going to form water when I do that, and I'm going to hook these two together in a very special type of bond between this carbon and that nitrogen. That's called a peptide bond, which is why the term peptide is so common in the nomenclature to describe different kinds of chains. So there's my dipeptide. Di meaning 2, peptide. 2 of these peptide bonds, basically, is what they're saying. And of course, there's my water. Now, if I wanted to break these two apart, going backwards, hydrolysis, removing the water, adding it here, or I should say splitting the water, and adding it here, and I get my 2 individual amino acids. Okay? So a peptide bond is that special bond that holds 2 amino acids together. Okay? So let's go ahead and get that into our notes. Amino acids are held together by peptide bonds. Okay. All right, so let's talk a little bit about diversity, because I have only 20 different kinds of building blocks to create all the proteins that our body is made of. Is that reasonable? Is that something that can produce a lot of diversity? And the answer is for sure it can. So let me give you an example of how diverse how many different proteins can be produced by just using 20 different amino acids and hooking them together in different combinations. So let's assume a chain of 300 amino acids. So a kind of a medium-sized protein. And it consists of 300 amino acids that are hooked together like a long train. And the question is, if I have 300 amino acids and I have 20 different building blocks, how many different proteins can I make that are 300 amino acids long? And the answer is, and let's emphasize this, with 20 different kinds of amino acids, I can get an astounding number or a huge diversity of different proteins. And the answer to that is 20 to the 300. That's 20 followed by 300 zeros. That is a huge number. So the sequence, the different sequences that I can produce of using 20 different amino acids and having only 300 in a chain, is the diversity of that is tremendous. So that just gives you an idea of the diversity, or the possible diversity that you can produce with just 20 different building blocks. Okay. The largest protein, by the way, so we talked about this, is an average-sized protein. The largest protein we've ever found in the human body is referred to as titin, and I'm just going to give you a number here. So the largest protein in the human body is, roll drum, titin. And we're going to talk about titin when we look at muscle tissue. And titin consists of 10,000 amino acids. A very long chain of amino acids. Okay. All right. Okay, so let's talk a little bit about the structural forms that proteins produce. So the structure of proteins. And keep in mind, when we talk about structure and different kinds of structures, what we're really looking at is functionality, right? Form follows function. So it turns out there are 4 levels of structure to proteins. All right, and let's go ahead and identify what they are. Number 1 is going to be primary structure. That's what we refer to, that first level of structural complexity, is the primary structure. And what this is just the sequence of amino acids. So we're simply going to make a chain of amino acids, string them together, and that pattern that we produce is going to be the primary structure of that protein. Okay. The second level is called the secondary structure, oddly enough. And the secondary structure is a twisting and folding of the primary structure. So, and this is going to come in two forms, two basic forms. Number one, an alpha helix. We're going to take a look at some pictures in just a little while. Let's get the information down, and because this is a terribly visual idea, so we do need to look at some pictures and understand this. But right now we're just getting our notes together. So the alpha helix, and the second basic form is what we call the pleated sheet. The pleated sheet. All right, so that's the basic secondary structure, and as you might imagine, that alpha helix is that very famous shape that is often associated with DNA. So it's that spiral staircase appearance. That's my alpha helix. Okay. So we're going to take that long chain, which is our primary structure, and we're going to form that cycle, that helix, that sort of spiral staircase effect. The pleated sheet is a bending, is literally something that looks like this. So that long chain will simply create what looks like stairs. And let me very quickly demonstrate this using a sheet of paper. So I've got a sheet of paper here, and I'm just going to very quickly fold it. And if you just give me a second here, I'll do this. And here is my pleated sheet. Okay, and you can see that pattern being produced as the way the interaction between amino acids forms this pleated sheet appearance. Okay, so that's my secondary structure. The third form is the tertiary, or this third level of structural complexity is the tertiary structure. And here at the tertiary structure, we're going to take the primary and the secondary structure and just fold it up and create this like complex knot. We're just going to start bending it back on itself and just create this very complex knot-like structure. And that's going to be our tertiary structure. So this is going to create a three-dimensional by folding the primary and secondary structures in on themselves. Okay. So I don't know if you've ever played with this, but when I was a kid, there was a little toy. It was called a Slinky, and it was a spring, and when you pulled on a spring, you get that helix appearance. And if you weren't careful, you would end up with that in a big giant knot. It would be all twisted around and create a three-dimensional structure, and that's kind of what we're talking about here, is that kind of structure. So I would take something like this. Here I have my helix, pleated sheet, and then another helix, and then pleated sheet, and then another helix, and straight, and then pleated sheet, and then another helix, and I create this three-dimensional shape. using the first two structural first two structural levels to create a three-dimensional object. And this is where the protein has its function. It's at this level that we see the actual function of the protein emerge. What is that protein going to do? What job does it have? It's based on this three-dimensional shape that we see right here. So if you destroy this three-dimensional shape, then you're going to lose the function of that protein. Okay. So the first two layers are setting the stage, and then third layer, as we fold all of this together to create a three-dimensional shape, provides the actual function. Is it an enzyme? What is it doing? Is it transporting oxygen? Is it a molecule of hemoglobin? That function comes out of this three-dimensional shape that we are just talking about. Now I'm going to draw a line right here. I'm drawing that between the third and the fourth structure, or third and fourth level of structural diversity, because not all proteins have this fourth level. So here, I'm going to put that 4 below that blue line, just to emphasize the fact that not all proteins, most proteins are up here, first, second, and tertiary levels, and that's where they stop. But some proteins go on and create a fourth level of diversity or structural complexity, and that, of course, is called the quaternary structure. And that is where two or more polypeptides, and we've defined that term so you now know what we're talking about, form a single protein. So we're going to get this plus another one, or maybe even up to 4 or more, combined together to create one very large complex structure. Okay? And an example of that would be hemoglobin. And I just so happens that I have a nice little image that will help us understand this fourth level of complexity. So let's turn to that right now and take a look at that. And then we'll come back and we'll look at the other three together. Okay. All right, so here, and let's do it right side up. Here is a molecule of hemoglobin. Right, so this is the other. And hemoglobin is actually composed of 4 different polypeptides. So I have one right here, I have one right there, I have one right there, and one right there. So these flying saucer-like discs is called a heme group, and that carries the iron, and that iron is what binds to oxygen. All right, but the molecule hemoglobin is composed of 4 polypeptide chains. 1, 2, 3, 4. 4 of them combine together to create one large molecule that we call hemoglobin. That's an example of the quaternary structure that we just described in our notes. And of course, that protein is found in red blood cells. We'll talk more about that in lab when we do an experiment using blood to demonstrate a process called osmosis. So that's the quaternary structure. Let's go ahead and review the other structures. So I drew some pictures. They weren't the best pictures in the world. So let's go ahead and look at what a real artist can do. So here's my primary structure. It's a series of amino acids hooked together into a long chain. That's my primary structure. Then I'm going to take this chain, and I'm either going to form this helical structure that you see right here, the so-called spiral staircase, or create a pleated sheet that you see here, and that would be my secondary structure. And then I'm going to take this plus parts of just the chain itself. So not this entire thing will form these shapes here. So there will be some straight areas and then the pleated sheet, or some straight areas and then the alpha helix. And what I'm going to do is I'm going to combine that together to create a three-dimensional shape. So here's my pleated sheet. I'm sorry, there's my helix. There's my pleated sheet. Notice that not the entire protein is forming one or two of these things, but they are present. And by folding up on itself, it creates this three-dimensional shape that we call the tertiary structure. And as I mentioned, this is where functionality rears its ugly head. In the protein, once we form this three-dimensional shape, that protein is ready to go and to do whatever job it's designed to do. Some proteins take on this shape, which is the quaternary structure, which we talked about, where we have two or more polypeptides forming a single large molecular protein. And this one then is the functional form of that protein. So I'm going to take two of these, combine them together to create one molecule, and that is where the functionality comes into play, is when they're combined together. So individually, they would not have functionality. They need to become together to form this structure before functionality is imparted to the molecule or to the protein. But most proteins stop right here and get their functionality at the tertiary structure. Okay. Oakley dokleys. Let's move on and let's talk about enzymes. So enzymes. One term we can use to describe an enzyme is it's a biological catalyst. So that's two terms put together to describe what an enzyme is. A catalyst speeds up a chemical reaction. But is not itself changed in the reaction. So it's not a player in the sense it is going to be changed. It's going to be structurally changed. It is the same at the end of the reaction as it was when it began, or when the reaction began. So that's the catalyst part. The biological part is that this is a protein produced by living organisms. You do not have to be an enzyme to be a catalyst. So there are many examples of catalysts which are not proteins, are not produced by living organisms. But if you have an enzyme which is acting as, a protein which is acting as a catalyst, then it is an enzyme and it can be referred to as a biological catalyst. Okay? So as I mentioned, these enzymes are capable of speeding up chemical reactions, and they're absolutely essential for life. Life could not exist as we know it without enzymes. So enzymes are extremely important in the functioning of your body, because without them, you couldn't exist. So it's speeding up chemical reactions. So many of the chemical reactions that you need to sustain life will happen whether an enzyme is involved or not, whether there's a catalyst involved or not. But the rate at which they would happen would be so slow you could not sustain life. And let me give you an idea of how important these enzymes are in terms of speeding up the reaction rate of a particular metabolic reaction in your body. So the range, or the range of the speeding up of reaction rates is between 100 million to 10 billion times faster than the reaction without the enzyme. So we're talking about a tremendous boost in the rate of reaction for a particular metabolic pathway of 100 million to 10 billion times faster than it would ordinarily take place. And with this kind of increase in reaction time, you can sustain life. Right? So let's take a quick look at the composition of enzymes. And here I'm not talking about the composition of proteins. We already know that. We already know that enzymes are proteins, and we know how proteins are put together. I'm looking specifically at enzymes. And it turns out that there are two parts to enzymes. There is the protein part, which is referred to as the apoenzyme. And there is another part, which is a non-protein part. Okay. So this is the non-protein part. And the non-protein part can be inorganic. Okay. In which case we refer to that as a cofactor. And it could be something like calcium is an example. Calcium is used as a part of an enzyme, and we would call that then the cofactor of that enzyme. Okay. Or we can have organic cofactor, which would then be called a coenzyme. And for us, many of the coenzymes, that is the organic cofactor that's part of the enzyme, are vitamins. So if you look at a chart listing the vitamins that we need in our diet, and you look at the function of those vitamins, by far, most of them will say coenzyme, 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 all the way down the list. It's amazing. And in fact, your textbook, I'm not sure what chapter it is. It might be in the chapter on nutrition, or it might be at the very back, at the very back of the book. But they do have a chart listing all the enzymes, all the vitamins that you need in your diet, and list the functions and the source and all that. And if you look at the functions, you'll see vitamin after vitamin is listed as a coenzyme. So very important. So how do you put all this together? How do you understand the relationship between the protein part of an enzyme and this non-protein part of an enzyme? And the way I like to think about that is the following. So imagine, if you will, you have a car, and the car is fully functional. It has a full tank of gas, fully charged battery. It's working perfectly. There's nothing wrong with it. And you get into the car and you want it to go, and you forgot your key. Well, the car's not going to go anyplace, right? Because you don't have that little last piece of part of the car, which is not actually part of the car, but fits into a slot in the car, and then the engine starts, and then the car becomes functional. So the protein part is the car, and the non-protein part is the key. You have to have that key to start the car. You have to have this either cofactor or coenzyme present and bonded to the enzyme before it works. Without it, it's not going to work. And that is why vitamins play such a vital role in your health, because if you don't have the vitamins, many of the enzymes that you are composed of will be non-functional. You'll build them, you'll have the protein part, but you won't have the coenzyme, and they won't function. They will be that car without the key. So that's how I think about this, is you need both. You need to have a functional car, and you need to have the key to start the car. You need to have a functional protein, and then you need to have the cofactor or the coenzyme to start the protein so it can function the way it's supposed to work. So how does an enzyme work? What does it do to speed up the rate of the reaction? So enzymes speed, as we've just demonstrated, up the reaction rate. How does it do that? What does it do to actually speed up that reaction rate? And here in this chart, we'll see how that works. So let's take a look at this. Put this aside. So this is a graph. It's a very simple graph. And here on this axis, we have potential energy. And along this axis, we have progress of the reaction. So this is time, and this is energy right here. And we have the reactants, and we want to get the products. Okay? Now, in a normal reaction, without an enzyme present, you would have to put in this additional amount of energy right here to get to this place right here. In other words, you would have to put energy into the reaction to make it go forward, so you would get the products. And what the enzyme does is it lowers that activation energy. So the amount of energy you have to put into a reaction to make it go forward is called the activation energy. And enzymes lower the activation energy, and so that can be driven forward. So notice that in this condition, if we have an enzyme present, that reaction will happen without adding any additional energy. There's enough energy in the system right now to drive that reaction forward if we have an enzyme. If we don't have an enzyme, we have to put in some additional energy to get it to go forward and have the end products. Okay? That is how enzymes work, is they lower the activation energy of a reaction. Okay? So, coming back to this. Enzymes lower the activation energy of a reaction. And that's how they work. Okay? Now, when we look at a particular enzyme and we observe how it's lowering the activation energy, this is the process by which that's all going to take place. Okay? So, if you look, here we have an enzyme, and this enzyme is referred to as sucrase. So here's something that you should pay attention to, the ending, ASE. Whenever you see a word with that ending on it, it's telling you that I'm an enzyme. So sucrase, it's telling you I'm an enzyme because I have this ending, ASE. So that's my enzyme. And with every enzyme, there is a so-called activation site. And this is, you can see here outlined in blue. So that's my active site. And what's going to happen is the substrates, which notice are both sucrose, that's a disaccharide, and water. And remember that what we're going to do is a dehydration or a hydrolysis reaction here, because we're going to split that water apart and we're going to create two simple sugars from that disaccharide. So this thing is going to lower that activation energy, and here it is, the products, glucose and fructose, and here's my water connected there. That's what we split out. And then the enzyme, you notice, has not been changed. So the enzyme is doing this millions and billions of times in a second. As long as there's substrate present, the reaction will go forward using that enzyme. So the way I kind of think about this whole idea is when you go to the store and you've spent the last few hours shopping for the perfect pair of pants, And finally, you find the perfect pair of pants after you're exhausted now, and you put them on, and they don't quite fit. They're almost there, but they're not there. There's a little tension around the belly. And so, what are you going to do? Well, if you're me, you're going to realize that I've just spent 3 or 4 hours trying to find a good pair of pants. I found them. They're a little bit tight, so I'm not going to give up. I'm going to buy them and go on a diet. So that tension around my waist is causing me to change my behavior. That's kind of how I see this. So there's a tension in this bond right here between the two sugars, between the glucose and the fructose. And that tension, as a result of that molecule sitting in this active site, is going to be enough to break that bond and split water and come out with my product. So that is how enzymes are working in our body. All right, so lowering the activation energy of a reaction. That brings us now to the last area that we want to discuss, which is nucleic acids. And what we're talking about is DNA, which is deoxy, ribo, nucleic acid, and RNA, which is ribo, nucleic acid, and nucleic acids, or these molecules, DNA and RNA, are composed of nucleotides. And nucleotides are composed of 3 parts. 1, a sugar, and that sugar is going to be deoxyribose. Or ribose. A base and a phosphate group. Okay. So let me go ahead and pull out a picture and take a look at take a look at this. So here's my phosphate group. There's my sugar, and there's my base, and that is a nucleic acid. Okay. Phosphate group, sugar, base. Phosphate group, sugar, base, so on and so forth. And that's all we need to know. And of course, it forms, in the case of DNA, this double-stranded double helix. This double helix. It's a double-stranded molecule. And really, that's all we need to know for this class about nucleic acids. Now, I want to talk about a very special nucleic acid, and that is ATP. So this will finish up our discussion here. So let's take a look at ATP. And ATP, if you don't know, stands for adenosine triphosphate. Triphosphate, 3 of those babies. I'll come back in here. There you go. All right. Let's take a look at the molecule. And I hope you'll notice something about this when we look at it. So I have a base, I have a sugar, and I have, in this case, 3 phosphates, not a single phosphate. So I have a phosphate group, I have a sugar, which is ribose, and the base. So this is a modified amino acid because it has the sugar, the base, and the phosphate. But in this case, if you look carefully, over here we have 3 phosphate groups. And if you look even more carefully here, you will see that this line is not straight. It's a little squiggly line. There's a squiggly line here and a squiggly line right there. That little squiggly line means that this is a high energy bond. And the reason it's a high energy bond is I've taken this, which is a phosphate group, it has a negative charge, and I've attached to that a second phosphate group, which also has a negative charge. And if you recall, negative repels negative. It's like trying to get the south pole of one magnet connected to the south pole of another magnet. It's going to be almost impossible for you to make those two things touch. But in this case, we did it. So we had to put a lot of energy into this bond in order to create that phosphate, in order to create this bond between that phosphate and that phosphate. And then on top of that, we take a third phosphate group with another negative charge, not only 1, but 2 negative charges, and we're hooking it to the 2 existing phosphate groups. That is a super high energy bond. So this is even more difficult to do than this. But it gets done. It gets done in our cells in a structure we'll talk about in the chapter on cells called the mitochondria. So we've connected now 3 phosphate groups together. We have 2 high energy bonds that are connecting the last 2 phosphate groups. And that means if I break this bond and release this phosphate group, I'm going to get a lot of energy released. And I can use that energy for work. And that is exactly why this molecule is so important for us, is because by creating this molecule and then breaking that bond and releasing that phosphate group, I get a lot of energy out of that, and I can use that energy to do work in my cells. And that is why ATP is in fact, the energy currency of the cell, is because it's put together in this fashion. So adenosine triphosphate has 2 high energy bonds. When broken, release a lot of energy, and that energy is used to do work in the cell. So ATP, adenosine triphosphate, and that's why you will talk about forevermore ATP in a variety of different instances, in a variety of different systems. We'll talk about it a lot when we get into muscles, and because we'll need a lot of ATP to drive a muscle contraction. So, ladies and gentlemen, we've come to the end of this lecture, and we've come to the end of the lecture in general on Chapter 2, which is the chemistry. And the next chapter we're going to visit is Chapter 3 on cells. And so thank you very much for your time and your patience and your efforts. I will see you next time when we begin talking about cells and their structure.